The focus of this short video is to be familiar with the fundamental autoantibody concepts, which we may come across in serology, clinical hematology, clinical chemistry, and blood banking. Do you know how autoantibodies arise, which may cause these diseases and syndromes? How do these diseases occur? And we will go over a few basic fundamental concepts of the development of autoimmune disease, and in later videos, go over the specific autoantibodies as they pertain to the clinical lab. Later, we'll focus on answering a few questions similar to the ones in the ACP Board of Certification Study Guide. What is an autoantibody? An antibody that is produced against an antigen found in an individual's own cells, tissues, and organs. As T cells mature in the thymus, they differentiate to cytotoxic, helper, and regulatory T cells. Cytotoxic and helper Ts encounter self antigens and develop self tolerance. Cytotoxic T lymphocytes are also known as CTL, T killer T cell, and in flow cytometry, specialists gate fluorescent signals in cell populations known to have cluster designation CD8 positive. Natural killer cells, also known as NK cells, are a type of cytotoxic T cell that is large and granular and is geared for nonspecific killing of virus infected cells and tumor cells. Natural killer cells function as nonspecific killing cells of tumor cells and infected host cells using enzymes perforin and granzyme as their main weapons. T helper lymphocytes are CD4 positive. Cytotoxic and helper Ts help coordinate a defense against infections and other foreign substances. They encounter self antigens and develop self tolerance. Antigen presenting cells are special macrophages, B cells, or tissue dendritic cells that present antigens to T lymphocytes with T cell receptors. Antigen presenting cells have the major histocompatibility complex that recognize self antigens and present them to the T cell receptors of T lymphocytes. Antigen presenting cells recognize foreign invaders with their foreign antigens, and they have major histocompatibility complex on these APCs. They become antigen presenting cells to naive T cells and sort of educate them to become mature T cells that remembers and recognizes these antigens in the future. This plays an important role of the cellular adaptive immune system. T helper cells secrete cytokines such as interleukins that signal other immune cells. TREGs are T regulatory T lymphocytes, and they are a type of white blood cell that help regulate the immune system with anti inflammatory properties, preventing the body from attacking its own cells, thereby limiting autoimmune disease. TREGs is a subset of CD4 positive T cells, also modulate T and B cells, neutrophils, monocytes, and antigen presenting cells. But those defective T cells that express T cell receptors with a strong affinity are recognized and should be destroyed in two phenomena known as negative inhibition and negative selection. Normally, TREGs modulate and inhibit cytotoxic and helper T cells that are detected to be overactive. This is immune negative inhibition. TREGs were formerly known as suppressor cells because they are known to be immunosuppressive. TREGs also regulate other immune cells by secreting interleukins. Most of the cytotoxic and helper Ts undergo cell death known as apoptosis. Programmed selective apoptosis of these defective lymphocytes is an example of immune negative selection to get rid of these autoimmune cells. However, if apoptosis does not occur with these defective T cells and they escape from the T regulatory cells, then autoimmune disease may occur. Then defective overactive B cells produce autoantibodies. Polyclonal activation of B cells is an adaptive immune response to infection where foreign antigens are recognized and attacked through its many different parts with different immunological epitopes, thereby activating many B cell clones. Heterogeneous polyclonal antibodies ensure elimination of most foreign antigens. Unfortunately, the collateral damage of this strategy is the cross reactivity with host tissue antigens. Complex genetics with the major histocompatibility complex and human leukocyte antigen plays a role in autoimmunity as we see in family and twin studies with a susceptibility to autoimmune diseases. FYI, an example is HLA-B27, which is linked to ankylosing spondylitis, an autoimmune disease causing inflammation around the spine. Sometimes cryptic antigens hide under the radar, coming out during tissue injury and are released, confusing the immune system to not be so tolerant. Hormones like estrogen influence the prevalence of autoimmune disease, as women are more likely to get autoimmune disease and at a younger age than men. An example is lupus, as we've reviewed in our other video. FYI, studies indicate that estrogen affects T helper cells that tend to get overactive. Prolactin is another hormone that has a stimulatory effect on our immune system. Studies show that stress with epinephrine, the fight or flight hormone adrenaline, has been linked to autoimmune disorders. An example of an environmental cause is in lupus. Ultraviolet light causes DNA damage to the skin cells, making them targets for anti nuclear antigens, stimulating anti double stranded DNA, anti SM, anti ribosomal proteins, and others, and causing the distinctive butterfly malar rash. Also, organ and systemic manifestations. And now for the questions and answers, which are similar but not exactly like the ones in our ACP study guide. This introduction to some basic concepts of autoimmunity will hopefully lead you to further studies on the pathophysiology of autoimmune disorders. Please follow us for more videos on autoantibodies, where there are many questions in the ACP study guide.